Hello, everybody. Thank you so much for joining us here today for Research America's Alliance discussion. And we really thank you for your partnership with Research America. If your organization isn't a member, please get in touch with my colleague, Joel, whose name will be in the chat, and he'll be able to give you information about the pros about becoming a member of the Research America Alliance. And I'm really happy to say that our presenter today is Dr. Adam Felsenfeld, who is the program director for the Division of Genome Sciences at the National Human Genome Research Institute. And he is going to be speaking to us about a new program, and I'm going to let him explain it, but I will say it's molecular phenotypes of null alleles in cells. And it's a new program that is just getting off the ground and is very exciting and holds a lot of potential promise. And as we always do, I think, please put any questions you might have in the Q&A box or the chat for the Zoom call, and then we can try to get to your questions during the Q&A session. And I just want to thank Dr. Felsenfeld so much for being here, and I'm going to send it over to him.
Great, thank you very much. And thank you, Sheila and Samantha and Ellie, for the invitation and for the support. My name is Adam Felsenfeld, and as Sheila said, I want to talk about a new program called Molecular Phenotypes of Null Alleles in Cells. And before I start, I do want to acknowledge that the other folks here who have worked on this program, Colin Fletcher, Ajay Pillai, and Riley Wilson. And before I get into this, I want to just sort of frame it by letting you know what my job is, because this description up here in the title, unless you're familiar with our bureaucracy, it doesn't tell you very much. So I do want to give you a sense of what my job is, what the job of a program director is. So Congress gives NHGRI part of an appropriation. Most of that pays for grants to independent investigators. And almost always, an applicant from a university, for example, a scientist will send in a grant proposal based on an idea that they have. It will go to a scientific review, and if it does well in review, it will get funded. And in part of this process, it will get routed to an appropriate institute at NIH. If it's about genomics, it will often get routed to NHGRI. And then once it's funded, the program director evaluates award progress and adherence to policy, and we have influence on funding level and some other things. And program directors usually have a portfolio of grants within a broadly related topic, and we have to maintain knowledge in the scientific area. And all this is sort of routine. We do strategic planning. We propose coordinated program concepts to address gaps and opportunities identified by that strategic planning, and we manage the resulting programs. We manage them towards goals and milestones, deliverables. We help develop standards. We deal with human factors, because these are mostly run as consortia, interpret policies or make new policies that are appropriate to the science. And we facilitate collaborations within the consortium and between the consortium and collaborators, and we manage change. And change is a constant in genomics. The technologies are always changing. There's always a better way to do something coming up. It's very exciting, very fast moving. So my description of the program today is going to reflect each of these steps: strategic planning, proposing the concept, and managing the program, just as an outline. Note that this is all still managed through grants and contracts to outside scientists. Program directors do a lot of things, but we don't do the actual experiments, and we don't get directly involved in producing or working with the primary data. We don't do the primary data interpretation. This is up to the scientists we fund. So back to Morphic, and I'm sorry about this URL, but if you went to that URL, you could find the program. But if you go to the NHGRI website and type in Morphic NHGRI or Google Morphic NHGRI, you'll find the website describing the program so far. So please note that the awards to this program have only just been made about a month ago. So I can tell you the origins and the rationale, the structure and the data types we expect, but I can't give you any results. But I hope by the end of this talk, you will understand how this program was created, what in the world molecular phenotypes of null alleles in cells actually means, and why it's important. Excuse me. So for describing the planning, I'm going to start with some simple questions. So genome sequencing is now extremely cheap. Millions of individuals have been sequenced. Tens of thousands of organisms have been sequenced. But what does a sequence mean? So interpreting it is not straightforward. Wouldn't it be great to know what each and every gene does? And wouldn't it be great to learn or predict what variants or mutations in each gene will do, what effect they'll have? This is really essential for understanding a key part of biology, and that is going from the sequence, the genome sequence, to the final form and function of an organism. So that's going from the genotype to the phenotype. I'll talk about that a little bit more in a bit. This will help us understand disease risk that results from gene variants, and could give us clues for how to intervene, and it may do some other stuff, which I hope to describe. Just to give you a feeling for phenotype, this is an example of genotype to phenotype in mouse. Here's a phenotype called polydactyly in mouse caused by a mutation in a gene called ALX4. This is taken from the paper cited in the lower left-hand corner of the slide. And I want to draw your attention to the row labeled plus plus. That's a normal phenotype, forelimbs and hindlimbs. They have the normal number of digits. And the bottom row, that's an ALX4 mutant. And you can see it has extra digits. So usually the key points are that the word phenotype can cover almost any aspect of form or function, though usually refers to anatomy, physiology, or behavior. Usually refers to organismal phenotypes, though not always. Note also that phenotype applies to the normal condition and also the mutant one. Finally, phenotypes can be striking with a large effect, like this mouse example, or they can be really subtle, hard to see, or not appear in every individual with a mutation, or otherwise more subtle than this. By the way, I should point out that this is not the only gene in mouse that when mutated leads to this phenotype. There are perhaps 15 others of them, which should already be telling you something interesting about gene function. So back to this slide, these are important questions for genomics in particular, and more generally for much of biomedical research. So it's why these general questions drive so much of the NHGRI 2020 strategic plan. In 2020, NHGRI published a new strategic vision based on input from the scientific community taken from several dozen meetings held over 2 years. And here's some excerpts from that plan. Right here, it says we need comprehensive views of genes and regulatory elements, starting with establishing the function of each human gene, including the phenotypic effects of human gene knockouts. So it's right there in our vision plan. The vision document also makes some bold predictions, including this one, which just the first part of that sentence, the biological functions of every gene will be known. And you'll see this is bold predictions for human genomics by 2030. It's great to have deadlines, but 7 years from now seems pretty short. In the case of the planning phase for Morphic, so a lot of the groundwork was just laid when we did our strategic planning meetings for the 2020 vision, with the community identifying the opportunities. But this is generalized at NHGRI. Almost whenever we have an idea about something, a program, we convene the community and ask them about it. So now that the planning part of the talk is covered, on to the proposal. And please realize this concept proposal is a pretty close match to what I proposed to our National Advisory Council in getting approval for this program. So you know a little bit about the process. So how do you go about learning these things, getting the answers to these questions? So you can look, there's actually a number of ways you can look when and where the gene is expressed in the organism. You can study the proteins it makes. You can study their structure, location, the amount, what other proteins they interact with. There are a lot of ways, and they're all important and complementary for full understanding. But sooner or later, you're going to have to just go in and break stuff, of course, in a well-characterized and thoughtful and organized way, and look at the effects. So people in genomics sometimes generally refer to this kind of study as perturbation by function or just a perturbation study. Before the genomics age, this used to just be called a genetics experiment. But when we can really do these at high throughput, I think they deserve a new category. So breaking things in a systematic way is just what Morphic proposes to do. We're going to create a catalog based on the following. We're going to make null alleles. These are essentially gene knockouts. I'll explain this in the next slide. Of every gene. That's the long-term goal for every protein coding gene. But we're going to start with a small number. So the next 5 years, 4 or 5 years, we're going to start with 1,000 as a pilot. And then look at the cellular and molecular consequences, the cellular and molecular phenotypes that result from breaking each gene. And we want to do this in human cells, in culture, and specifically in systems that are as close as possible to their actual native biological context, so differentiated into the multiple cell types. To unpack this a bit, so null alleles, this is an old genetics, null is an old genetics term that really means that the gene function will be completely knocked out. For background, please be aware that not all mutant alleles completely abolish function. Some just reduce gene function. Some can have effects that are worse than a complete knockout. But in Morphic, we're going to concentrate on null alleles for several reasons. First, they are expected to have strong, readily observable effects, so easier to detect. We know how to reliably engineer them into human cells. We only have to look at about one knockout per gene. That's about 20,000, so it's not too big a job to try to test every gene. Fourth, null alleles are useful for interpreting other alleles in the same gene, so weaker losses of function, or interpreting similar phenotypes seen for other genes. So that's the mouse example I showed you where I said that there were 16 genes that cause polydactyly, loss of which caused polydactyly, tells you something about function. The results also will be complementary to extensive knockout phenotype data, organismal phenotype data from something called the Knockout Mouse Project. I'll talk about that a bit more later, which has anatomical and physiological and some behavioral phenotypes, but very little molecular or cellular data. I think I missed something here. So molecular and cellular effects or phenotypes. So this is potentially everything from gene expression, so is the gene active, when and where, to cell metabolism, like what lipids it's producing, to the cell's developmental potential, to cell shape, and on up to cell physiology, like presence of various ion channels. This is the output and perhaps the most important part to get right. We want the tests of molecular and cellular effects, or assays, I'll call them assays, or effects, or tests of phenotype, to be as informative as possible. For that reason, Morphic will be done in human multicellular culture systems, ideally cell culture systems that recapitulate important features of human organs or early development. This means we can get information about interactions between cells, cell-specific functions, and organ or tissue formation. It also means it will be possible to do assays across multiple cell types in the same experiment, so to multiplex, and to basically capture as many functions as possible in an assay. More and more work is being done with these organ-like systems in cell culture. You may have heard about them. They're called the organoids, and they can be derived from biopsied samples from people, from induced pluripotent stem cells, from embryonic stem cells. A lot of work remains to be done to improve these methods, to get them to faithfully develop into things that are more experimentally like human tissues. But they're enough at this stage, far enough along to pilot a program like Morphic. And in the next 5 years, I expect that the technology for organoids will improve a lot. There's a lot of work going on there. So some general benefits of Morphic. So we're missing functional information for many genes, and Morphic seeks to establish a consistent base level for all. to try to help the entire community. It won't replace everything, won't replace individual experiments, but it will raise the floor of the place where people are starting. Even for genes where we do not know the associated anatomical and physiological phenotype, and where we have some molecular data, there's usually a gap between the two that we don't understand, and Morphic will provide data that can help fill that gap. Third, it's a foothold. The data will be a foothold to understanding mechanism. So genomics, a lot of genomics data is correlative. It's discovery data, it's large data sets, and then it's like correlations between a variant and a disease or susceptibility to a disease. So correlative, but we don't want to stop at just correlative, and we want to begin to get sort of high throughput insight into mechanism, and filling in that gap with molecular data between genotype and phenotype with molecular cellular phenotypes, we think will help. Finally, these data and finally, the cell lines that result, these are potentially, you know, a cell line, a stem cell, human stem cell line with a mutation, with a known mutation in a known gene. And each of those is potentially a model for more detailed study. I wanted to provide a kind of higher level conceptual view of what we hope Morphic can do. I'm not sure this captures it. I've experimented with this a bit. This diagram illustrates layers of the things that can be measured or assayed that lie between the DNA sequence at the bottom and organismal phenotypes like physiology and anatomical features, including, for example, genetic diseases, at the top. They're at organismal phenotype. To begin to get the idea of what an eventual catalog might do, imagine a perturbation at the bottom DNA level. This could be a sequence variant, occurs naturally in the population, or it could be a mutation that was induced. This would often have consequences throughout all these layers on up through this diagram. There would be correlations of each perturbation with the gene expression, with what's happening with proteins and other molecules, what's happening with the cell shape, cell survival, you know, what the insides of the cell organelles look like. It would have influence on how organs and tissues are organized, all the way up through organismal phenotype. And if we can fill in enough of these, we think this will be incredibly useful for understanding phenotype. If we can make enough connections in a systematic way, we might be able to use the information to accurately predict missing data, so missing links between the layers. Given the enormous scale of this problem, it looks like there will always be missing data. To get an idea of the scale of the problem, if we actually wanted to do real assays of every one of these things, it would be on the order of billions of assays. So that's the number of genes times the number of assays times the number of cell types, just to begin. So that's billions of individual experiments, and we're nowhere near being able to do that now, and probably not in 5 years, and maybe not in 20 years. But if we can fill in enough of these in a logical way, in a systematic way, I think we will be able to start predicting other links that exist and then testing for them. So there are challenges. Can we knock out every gene? I'm pretty sure we can do this. A lot have already been knocked out. There are already cell lines with known knockouts that cause lethality. So a lot has already been done. The second big challenge is, can we do enough informative assays or tests of cellular and molecular phenotypes? And do we have the right assays for the right cell types? And that's really hard to know. I really think that if we can get at least one repeatable, consistent molecular phenotype for each gene, even if it doesn't seem it's like the right one for a particular function, but it will tell you something. I think each one piece of the information will be useful, and I do think that it will converge. Another, I think this is the biggest challenge of the pilot phase, is to see if this is true. Another challenge is that these null alleles, these strong alleles, may actually kill the cells, and those are really hard to interpret, or have multiple different effects on different cells. That's hard to interpret. And there's always going to be a lot of biological variability in a system. It could be genetic background. It could be, how the cultures are grown. And we are not going to be able to exhaustively answer that question, but we should be able to get a good sense of how bad that challenge is, because that biological variability actually could really set things back if it's extreme. Morphic depends on other function studies. Here's that diagram on the left that I showed you before, and here are a number, not an exhaustive list, of other studies. So up at the top is the Knockout Mouse Project, which has the phenotype data, along the organismal phenotype data, along with the knockouts. There are other initiatives like it's the, sorry, I always forget these, excuse me, genotype. Why did I go back? The genotype to expression consortium data, which has gene expression by variant data, and others. And you can see that Morphic fills in certain levels of data that the others are not as strong at, and the others fill in levels of data that Morphic isn't as strong at. And if this really works, and if we get, for example, if we make the data so it can be truly interoperable and computable, and all these others can as well, we'll have quite a powerful resource. So that's that's sort of the concept proposal. That's the concept of the program. And following that, the concept was approved. We issued funding opportunity announcements to the community, and we made a number of awards for data production research and development centers to Paul Robson at the Jackson Lab, to Danwei Hongfu at Memorial Sloan Kettering, Mazar Adli at Northwestern University, and Luke Gilbert at UC San Francisco. We also made a Data Resource and Administrative Coordinating Center Award at the University of Miami to Stefan Schurer. We are still under review is the RFA for data analysis and validation centers, who will actually get the first crack at trying to combine these data and use them to basically show that they can be used to answer important questions. The data production centers will pursue roughly a dozen different assay types, depending on how you count them, in different organoid or similar systems that will model brain, heart, and early development of tissues derived both from stem cells and some from primary samples. And this is just a little bit of what we have to do to even have a chance of making an argument for a phase 2. Is basically to show that high quality data can be produced at scale, make the null alleles, test multiple mutagenesis methods and assays to see which is the best, get the data QC and the interoperability down, start looking at comparisons both between different centers' data, and also swapping, for example, cellular systems between the data producers so they can run assays on each other's samples. Think about prospects for throughput and cost improvement. Until we've defined what the bottlenecks are, it's really hard to do this. We are inevitably going to find out that our scientific challenges list, we're going to have to worry about things that we didn't anticipate. That's going to happen. And get some of that data, initial data, analyzed, show that it's useful, and get it out to the community. So that's the task for the next roughly 3 years. And with that, I'd like to wind up and ask for questions. I want to thank my colleagues here for input on ideas for this presentation, including Ajay Pillai, Colin Fletcher, Riley Wilson, and our division director, Carolyn Hodder. And that brings me to the end of the presentation.
Great.
So thank you.
Thank you so much. What an amazing area of growth and development, really exciting. I have a couple of questions to kick off. You talked about that the initial subset is of genes that you're going to focus on is 1,000. And do you specify which ones are included in the 1,000, or are the folks who get the grants deciding which genes they would like to focus on? How does that process work?
Yeah, it's a little bit of both. So we don't want to, what we want programmatically out of this is enough genes of enough different supposed function, sort of hypothesized function, or a little bit known function, to have a good spread of sort of different types of genes that are active in different tissues, different stages of development, in order to get a sense of what a larger sample will look like. And this is modeled after other projects, like older projects where what was initially too expensive or impractical to go after the whole genome. So pilots picked 1% of the genome. So this is very much like a pilot that picks 1% of the genome to look at. And when those other projects have been successful, they've quickly led to whole genome efforts. But we didn't want to tell people which ones, because it really depends on their assays. So those two things are linked. But it can't be completely open, because then we won't be able to coordinate. We have to understand how to coordinate. So one of the things we're going to do now that grants are awarded is talk about gene lists and priorities and look at overlap, and try to get a good spread and an efficient design across the consortium.
And going back to your earlier slide about the different anatomical functions in the mice, you said there were 16 different genes, I think, that could affect the polydactyl expression in a mouse. So does that mean that we already know what the knockout effect would be of eliminating those genes, or how would we already know that?
So there are a few genes, especially ones that were identified over the years, over the decades, that have striking effects that scientists have worked on even before there was for example, a mouse genome sequence. They would, ever since people have been able to clone genes that or loci that correspond to a mutation, starting way back with cystic fibrosis and even before in other organisms. People have studied over years the effects of these things, and quite a lot is known about polydactyly. Actually, they know that there are a set of genes that encode positional information in the developing limb buds, and if that pattern is disrupted, the organism ends up with more digits than normal. But the other thing is that each of these 15 genes also has other effects, and the effects overlap some, but not entirely. So this whole story is much more complicated.
Like most things in life, I think.
And for most genes, we don't know. It doesn't have such a clear phenotypic effect. We don't really know. You might get genetic syndromes and things like that. It's not always so clear what's going on.
Right. And it seems like one of the major areas of research is not necessarily something that is a congenital genetic defect, but the effect of the environment or exposure to different chemicals or, different things throughout the life can affect the structure of the gene. Will both types of genomic dysfunction or malfunction or whatever be included in the study that you're doing with the 1,000 different, excuse me, I'm going to put you on mute for a sec.
Sure.
Apologize. Will that intersect with the the 1,000 set of genes that
Yeah.
you're looking at?
Yeah, so not directly. We're not thinking about environmental perturbations, but in the general terms in which I framed it, perturbation could include a chemical perturbation.
Mhm.
And it wouldn't surprise me to see that if a mutation causes a set of molecular phenotypes that results in an organismal phenotype downstream, that a chemical perturbation that did the same thing would affect the same process. And we should see that. And in fact, there's a lot of work that's been done like that. These are even before the genomics age. These things were called phenocopies. In fruit fly, if you hit a embryonic fruit fly with ether, it can develop an extra set of wings, for example, which recapitulates a phenotype of an important classical mutation in Drosophila. So, and there are other institutes that are very interested in projects like this. And I know NIEHS is very interested in kinds of projects. That's Environmental Health and Sciences Institute sorry about the initialism. And they're very interested in molecular
that's going to come out of Morphic might provide some insights into diseases where multiple genes likely play a role in the disease and how it presents. Alzheimer's, as an example. Yeah,
Morphic would have to work extremely well. I mean, that's right. That's the ultimate goal. It is to go from the biology to the disease. And the truth is, I don't know exactly how that's going to look. I can wave my hands. I think that once you identify a phenotype with and associate it with a gene, even if it's a highly polygenic trait, you're going to start getting into these pathways. And I think that it will be helpful to have that information in a catalog once you get there. And in fact, this is not in a time series. This is actually what people are already kind of doing. They do an association study where there's a statistical association between a set of variants that occur in people who are affected versus those who are not. And they try to assign those variants to a gene, to a nearby gene or a gene that the variant actually is in, although they may not actually know what it does. And then they try to look for the molecular functions of those genes. And if there's a catalog sitting there, it should give them a lot more clues. And there are already some catalogs of function. They're not completely, they're going to get better and better, and people use them quite a lot. So I do think this will be useful in helping things along, but I don't think it's going to solve the problem. Again, I don't think this is ever going to replace, this is going to be generating hypotheses and sort of preliminary catalog or preliminary results in that frame. And people will still have to go back and do the careful studies over time, focusing in on the right assays to understand what's going on.
Definitely.
Can you talk a little bit about the, how the research on the overall genome project has led to the point that we're at now, sort of the previous 20 years of research and how, obviously 30,000 foot level, but it just seems like there's been such enormous progress in this area, and, kind of how it's led to where we are now and where we want to go next.
I don't know how to give a short answer to this question, but I'll try. Even when the genome was being sequenced, even before, people knew that it was hard to interpret. They didn't know where the functional elements were. They didn't know what the functional elements were. If you were, interested in this area 22 years ago, 21 years ago, you might remember that there were sort of betting pools for the number of genes even, and it was, people said 20,000 and some people said 100,000, and no one really knew, so we didn't even know how many genes there were. We didn't know almost anything about regulatory regions of genes. We had a couple of examples that told us that it was likely to be very complex and worked out by individual labs concentrating on those regions over many years, sometimes over whole careers. But trying to develop a catalog of functional elements was really the first priority. And that resulted in a project called ENCODE, Encyclopedia of I can't remember. I'm sorry. Okay. Hope my colleagues are not on this.
I'm sure they'll excuse you.
Which was basically a catalog of functional elements of all kinds, not just genes, but of regulatory regions and other kinds of elements. Once you think about ENCODE in that series, you quickly get to sort of higher level function in two ways. And one, you want to know what all the regulatory variants are and how they control the genes that they're mostly near. They're not always near, but they're mostly near. And identify them, because you can imagine you do an association study for heart disease and you can end up with implicating a variant, and it's not in a gene, doesn't obviously disrupt the gene. So how do you characterize it? If we can in advance characterize all the non-coding regulatory sequence, then we have a prior we can look at, and that is great for a catalog. So that's the next stage of that sort of functional analysis, is to start breaking those non-coding elements and asking what it does to the gene expression. And there's a whole effort here focused on that. And then there's another level of organization, which is how the genes basically interact with each other, and what the downstream consequences are for phenotype and the mechanistics. And that's more of the morphic emphasis coming in there. So that's sort of the, if you I don't know if any of you play civilization builder games where you have a tech development tree. So this is like a logical tech dev tree in real life.
Yeah, that's a very interesting way to think about it. Kind of apply it to sort of more general world situations. Sam, how are we doing on questions?
I think we're coming up on time, and we want to be respectful of your time. And I don't see any other questions from the audience at this point. So we'll ask one last question. We are an advocacy organization and a member organization. So how can advocates best support the work that's going to come out of Morphic and the work of the Institute?
Yeah, I think the best support we can have is continued support for basic research studies, but not just genomics, everything, because it really is all connected. And another thing is that I think that you probably have knowledge of and access to a bunch of different scientific communities as represented by your speakers. And having those people, especially communities who haven't spoken with each other, speak to each other, I think really can be very productive. And it's actually another fun part of my job that I like quite a lot, is connecting communities. I think there's a lot of good things happen when that happens.
I agree. Thank you. And thank you so much, Dr. Felsenfeld. This has been really terrific, really exciting new research, and appreciate your time today and your work on these new developments.
Great. Thank you so much for the opportunity.
You bet. I'm just going to make one quick announcement before everybody goes. Our next Alliance discussion will be Monday, the 7th of November at 1 p.m. And it's going to be with Aisling McDonough, who is Chief of Staff to Congresswoman Eshoo, who is the chairman of the Health Subcommittee of the Energy and Commerce Committee. So it'll be a very interesting discussion, I think, about some of the issues Congress will be grappling with in the near future. So thank you again, Dr. Felsenfeld, and thank you everybody for participating.
Take care.
Take care.